First, I thank the organizers for putting together this event. And our lab is about half a year old, still a baby lab. But today I'm going to tell you about some research from our group on the plant immune system, which arguably shapes a large portion of microbial interactions in nature or in your garden. So as most of you in this room know, that plants have an innate immune system, which is somewhat similar to our immune system. We share some molecules to detect, I think it doesn't work, but detect pathogens, and also we share some signaling mechanisms. And our immune system, so we have a suite of immune cells, specialized immune cell types, such as B cells, macrophages, T cells. They circulate around our body to eliminate pathogens. Plants, however, don't have any of these things. They do have a lot of cell types. You can find a lot of cell types in textbooks, but none of them are specialized and dedicated immune cells. Also, most of the plant cells are immobile because they are encased in a rigid cell wall. So basically, there's no police patrolling. But pathogens don't care, and they attack plants anyways. And this is the pathogen infection, how pathogen infection could look like in plants. This is a leaf infected by bacterial pathogen with GFP expressed. And point here is that pathogen infection can be quite heterogeneous. It looks almost like a random event. And this means that each plant cell doesn't know whether and when they will encounter the pathogen. So this means individual cells will have to commit to certain immune responses when necessary, while at the same time doing other fundamental jobs, such as photosynthesis or making plants bigger by dividing and expanding. So, and this creates a critical problem of who does what in the face of pathogen infection. And it's quite fascinating thinking about how individual plant cells make decisions and coordinate with each other to deploy a successful immune responses. So we also people naively thought that individual plant cells would respond in different ways, but we didn't really know how this might look like, because previous research mostly focused on the tissue level responses, grinding whole tissue up. But as Ian discussed yesterday, thanks to the development of single cell genomics technologies, now we can assess how individual cells respond. And that's something we're doing. So we use plant Arabidopsis leaf infected by bacterial pathogen as a model system. We infected plants with multiple strains. Some are immune activating, some are immune suppressive. We harvested plants at different time points. So the whole point here is to maximize the situations that plant cells might experience to really get a comprehensive idea of potential cell states. We isolated nuclei from those plants and applied single nucleus multi-omics, which allowed us to profile both transcriptome and epigenome from the same cell. For the epigenome, we specifically looked at the chromatin accessibility. As you know, chromatin can be either closed or opened. An open region can be bound by transcription factors and cofactors to regulate gene expression, so you could learn potential regulatory mechanisms. Now, here's the overview of the data we got. We've sequenced over 65,000 cells from a bunch of different infected plants. And this is a UMAP plot, which is the two-dimensional representation of very high-dimensional transcriptome data. Here, cells with similar transcriptome patterns are clustered together. And based on marker gene expression, we could identify major cell types, such as epidermis, mesophyll vasculature. But we can go ahead and identify further cell populations by subclustering individual major cluster. These are cell populations with distinct gene expression patterns. So we systematically identified over 400 subclusters, some of which might be meaningful cell states in plants. And here I'm showing about 20 famously known immune genes. we know that they are important immune genes, and you can already see the huge diversity of gene expression patterns all across those subclusters, just by looking at several immune genes. So this strongly indicates the existence of immune cell states, which is already quite exciting. Now, we have the single-cell multiome. We can directly compare transcriptome and epigenome. This is one example, just focusing on one gene, CBP60G. This is an important immune gene. What you can do is you can look at the chromatin accessibility all across different cell populations, and you can see a different accessibility in different cells. And you can also correlate this accessibility to the expression of this gene, which is shown in the violin plot, which may be too small to see, but in this case, they happen to significantly correlate. And then we can make a link between the gene and the chromatin regions. And in this case, this can tell you about potential regulatory mechanisms, right? Because some transcription factors might be binding to these regions to regulate this gene. And this can be experimentally tested in the lab. And we can do this kind of analysis genome-wide, transcriptome-wide, and identify thousands of genes that showed at least one significant link to the chromatin regions in at least one of those infection conditions. And for example, we found some defense genes are specifically linked to one or more chromatin regions only when infected by those two immune triggering pathogens, but not the immune suppressive pathogens or mock condition. This means that there is a coordinated changes of transcription and epigenome in the single cell or cell state specific level upon pathogen attack. So our lab is now following up on the actual molecular mechanisms of those potentially novel gene regulatory mechanisms. Now, what single-cell multiomics doesn't tell us is the spatial information. We have no idea about where those cells came from. And spatial gene expression information is critical to understand plant immunity or plant microbe interactions. This is a previous study using 2 reporter lines and then the live imaging of those promoter activities. So these are 2 defense-related promoters. And these regions are the pathogen-affected region. You can see a very different spatial and also temporal expression pattern of these 2 immune genes. And such difference cannot be captured by this rather static map of single-cell expression. So we do need spatial gene expression information, but we need more. There's more than 2 because we have hundreds of potential cell states and probably thousands of genes are involved in defining those cell states. So that's why we turned to another new technology called spatial transcriptomics, specifically a Murphy's technique, which is a massively multiplex single molecule imaging. It's a FISH-based approach that can visualize hundreds of genes at the same time on the tissue section. So we use, again, infected plant leaves and section in this way, the longitudinal section, which you can see here on the right. And we selected, so because this is a targeted approach, I manually curated 500 genes, including cell type markers and many known immune genes, also a bunch of transcription factors. We've done exactly the same time course upon infection of this immune triggering pathogen. And here, I'm showing only about 12 genes, but you can already see this massive transcriptional reprogramming upon pathogen attack with some interesting spatial structure. Here I'm showing one of 500 genes, ALD1, which is a known immune gene, and you can see a nice induction of this gene. This means that Murphy's is highly target specific. You don't see off-target signal from non-infected plants. So we basically have 500 of those images. So it's a bunch of images. And we further analyze all these image data to analyze the gene expression in a quantitative way and also at the single cell level. And this allowed us to analyze this image-based data pretty much in the same way as single-cell RNA-seq data, that's sequencing-based data. And this allowed us to integrate all the data from MERFISH, the image-based data, with the single-cell multiome, the one I presented earlier, to get the unified map of all the cells profiled by both technologies. Now we have hundreds of thousands of cells here. And we hypothesized that probably we can use the MERFISH cells as spatial anchors to spatially map the single-cell multiome information. So to test this, we've done a simple test where we try to spatially map those cell type labels predicted in the single-cell multiome data, just purely based on the gene expression information, no spatial information. And then we integrated this data with MERFISH and spatially mapped those colored labels, which resulted in this kind of picture, where you can see nice vasculature network in yellow, and then pretty much everything looks what we would expect. So the approach seems to be working. And this is nice because now we can spatially map any multiome information which contains whole transcriptome, not only 500 targeted, selected genes, but 20,000 genes. It also has the entire epigenome information. So this allows us to do a lot of things, and I will just show you one of the things we've done. So first, we used, again, this data-rich single-cell multiome data, no spatial information, to model the immune trajectory, which is predicted here, and I'm going to walk you through what it means. So one end of the trajectory in those cells, we think that these cells are either not immune activated yet or immune activated not so long ago. So we call them as young immune cells. And at the other end of the spectrum are older immune cells. They have been immune active for a long time. Probably the tip of the rightmost hand may be the cells that are initially infected by the pathogen upon infection. And this is a purely computational prediction, but we do have the real-time course data. And this supports our model, because you can see more blue cells early on and more red cells later. So it looks like we got the immune trajectory right. Now we can spatially map this immune trajectory based on the data integration with MERFISH, and this is what we got. So what you can see here is upon pathogen infection, you see the emergence of those immune active regions coming up, and they are growing over time. And then I think they merge together at the end and further changing their states. So now we could really model the spatial temporal dynamics of immune responses at the single cell level with the whole transcriptome and epigenome information. So having this map, we really wanted to find something interesting in biology, of course. And so we focused on what's there at the middle of those immune active hotspots, if you wish. So we zoomed in to one of the regions and found some interesting cell populations. So to cut a long story short, we think we identified two fundamental immune cell states. One is a cell that are in direct contact with the pathogen. We call them as primary immune responder cells, or primer cells. The other state includes cells surrounding those primer cells, so we call them bystander cells. So it's conceptually very simple, right? But they are fundamentally in different situations, and now we know the rich molecular information to differentiate them. For example, in the spatial data, you can see BON3, the yellow one, seems to be enriched in the primer cell population, whereas two other genes are more bystander cell. And completely independent single-cell RNA-seq data also showed that BON3 is enriched in a very rare cell population. This is a primer cell cluster. FMO1 and LD1 occupy different clusters. So this means there are at least two molecularly distinct cell populations, probably more than that. So we wanted to, in this particular study, we focused on the primer cell population because we found a lot of previously unknown genes enriched in this cluster. And we found an interesting transcription factor called GT3A. This is a trihelix type of TF And single nucleoside-seq showed that GT3A is indeed enriched in the primer cell cluster. Again, independent spatial omics Murphy showed that GT3A expression is very similar to BOM3, which is a primer cell marker gene shown in the last slide. Also, the single cell attack-seq, the epigenome data suggested that in the primer cell cluster, the GT3A binding sites are more open genome-wide. So those cells are ready to be bound by GT3A. So all in all, the data suggests that GT3A might be an important regulator in this rare cell population. And we do have additional data to support that GT3A actually contributes to the immune system. So the message here is that there's an advantage in having both single-cell multi-omic data and spatial transcriptome data, because this allows us to identify novel and potentially very rare cell populations, and you can go on and understand the potential regulatory mechanisms. So here's the model. So we identified 2 cell states among many other interesting cell states, primer and bystander. We hypothesized that primer cells might represent more on the cell autonomous immunity, whereas bystander cells might be more non-cell autonomous. There should be some signaling going on, which we don't know, and different molecular regulatory mechanisms are at play, which we are actively studying now. Kind of an inaccurate analogy to understand this cell population is that primer cells may be more like a smoke detector, which detects pathogen infection. And bystander cells may be like a sprinkler that actually splash water to extinguish fire. So we speculate that bystander cells may be the ones that are actively fighting against pathogens or suppressing pathogen growth. But I'm happy to be proven wrong. But the point here is that even without the specialized immune cells, plant cells can flexibly change their states and coordinate with each other to deploy this multicellular immune network. And I think we've just started to understand this, which is very exciting to me. Okay, so our lab is really interested in understanding immune cell states for their diversity, function, regulation, and communication. And based on, after getting a fundamental understanding of those, we might be able to engineer immune cell states in a specific way, very targeted way. So coming back to this analogy, if you don't want your house to burn down, you might not want to make your smoke detector to splash water or a sprinkler to detect smoke. You would want to improve each component in a more reasonable ways. And also, while improving this system, you don't want to mess up with your heater. And as I said, plant cells have multiple functions, and they are tightly linked. And actually, this is a real-world problem in agriculture. It's known as trade-offs. If you try to improve the defense responses, it can often come with the cost of yield, for example. So by understanding different components of the plants and do a targeted and accurate engineering of different parts, we may be able to stack multiple traits on top of each other. And that's something we'd like to test in the future. Okay, so I'm almost done, but I slightly regret that I didn't talk anything about microbes. But we are equally enthusiastic about understanding microbes and how they interact with plants. And here again, the single cell and spatial resolution is the key. And one of the things we're trying to do is to really visualize the entire microbiome at the single cell and single strain resolution, together with visualizing host plant responses. For this, we've recently developed a method called Phytomap, which allows highly multiplex in situ visualization of gene expression of dozens of genes. And so we are trying to extend this method to the microbiome members. So our dream project is to really visualize every single cell in this host microbiome, the whole host holobiome, to really understand how individual cells interact with each other. So hopefully I can tell you more about this at the next, when I stand in front of you next time. Okay, so these are the people who contributed to the work, and thank you for your attention. I'm happy to discuss over the coffee. Thank you.